I'd like to start thanking Marta Garrido for organizing this symposium and for inviting me, and then apologies for not being there in person due to health reasons. The talk is organized around two topics. In the first part, I will discuss evidence about affective processing in blindsight patients, which goes under the label of affective blindsight. And I will present some behavioral, functional, and structural MRI evidence. But I will also try to introduce some new clue, trying to move our level of understanding from the typical level, which is correlational, to a more causal level of understanding the neuronal basis of affective blindsight. And the second part, I will address visuo-motor integration to gain some clue about whether there is a role for the intact hemisphere in supporting blindsight. And I will present some data exploiting the Poffenberger paradigm to this purpose. Affective blindsight was originally discovered by Beatrice de Gelder, Larry Weisskranz, and collaborators in a behavioral study in 1999, when patient GY was demonstrated to be above chance level in discriminating facial expression into alternative forces choice task. Later on, we've demonstrated that patients with affective blindsight actually display emotional contagion, meaning that these patients have facial reactions that are congruent with the emotions displayed in the blind field, and also that these responses are faster for unseen than for seen stimuli. A number of studies in the 80s and 90s, primarily from Joe Ledoux group in rodents, have documented a subcortical pathway to the amygdala involved in fear conditioning, also without a contribution of the sensory cortex. So on this basis, we investigated 10 healthy controls in a DTI study, tried to see whether we could reconstruct fibers connecting the superior colliculus here in yellow, the pulvinar in blue, and the amygdala in red. And in fact, as you can see, we were able to reconstruct these fibers, which are represented in blue in the right hemisphere and in green in the left hemisphere. But we also try to examine whether the subcortical pathway undergoes plastic changes following early V1 damage. And therefore, we examined also patient GY with an early damage to V1. And we could see that fibers in the right intact hemisphere were present, but not significantly different from the fibers reconstructed in the same hemisphere of healthy controls, whereas the fibers present in the left hemisphere of GY, where he has a V1 damage, were higher, significantly higher in number compared to the number of fibers reported in the healthy controls. Finally, in this beautiful study, Marta Garrido and her group applied probabilistic tractography to more than 600 subjects and reconstructed again the subcortical pathway to the amygdala. But as we know, tractography does not provide evidence about a direction of information flow. So importantly, they computationally modeled information flow and found evidence for afferent pathway to the amygdala, but also they found that a greater fiber density is associated with stronger dynamic coupling between these three structures, and this coupling enhances fearful face recognition. After this short overview of some previous studies on the affective blindsight and its neuronal basis, I would like to present some relatively new data where, as I said, we try to gain some new insights that might support causal level of explanation about the functions of this subcortical pathway to the amygdala. To this aim, we exploited the fact that the superior colliculus receives mainly information from the magnocellular pathway that has lower spatial resolution compared to the parvocellular pathway that projects to LGN and therefore composes the geniculus striate pathway.
We filtered fearful and neutral facial expressions to display only high spatial frequencies or low spatial frequencies, as shown here, essentially using the same filtering parameters previously applied by the group of Patrick Viomier and Ray Dolan. And then we presented the stimuli in a two alternative forced choice task, asking participants to discriminate between neutral and fearful expression, and then after each trial to rate their confidence in the response just made on a 4-point scale, from least confident to the most confident.
With this design, we tested two blindsight patients, patient GY with a damage to his left V1 that resulted in right homonymous hemianopia, and to patient TN with a bilateral damage to the occipital lobe that produced complete cortical blindness. These are the behavioral responses, and as you can see, the patients display a very similar response pattern. In fact, both were above chance when asked to discriminate between fearful and neutral facial expression when they were in broad spatial frequency, so when the stimuli were unchanged, but they were also above chance when asked to discriminate between fearful and neutral facial expression when they were in low spatial frequency. However, performance dropped a chance when the stimuli were filtered to display only high spatial frequency information. As the patients rated confidence in each response on a 4-point scale, we were able to analyze behavioral responses with signal detection methods also. You see here the ROC curves for both patients, and it's evident that the perceptual sensitivity measured as D prime was higher than 0 only for broad spatial frequency and for low spatial frequency filtered stimuli, but not for high spatial frequency stimuli, meaning that the above chance performance displayed in these two conditions cannot be ascribed to response bias. Finally, you will see fMRI responses for the non-conscious perception of fear in low spatial frequency. And this contrast reveals significant responses in the superior colliculus in both patients, GY and TN, responses in the pulvinar bilaterally in GY and unilaterally in the left pulvinar of TN, and in the amygdala unilaterally in GY and bilaterally in TN. So overall, this finding suggests that the subcortical pathway to the amygdala involving the superior colliculus and the pulvinar seems specialized for processing affective information encoded in low spatial frequency, at least following V1 damage. And in fact, filtering stimuli to high spatial frequencies abolishes both the possibility for these two patients to discriminate above chance fearful from neutral expression and the corresponding fMRI activity. Now we move to the second part of the talk, where we'll discuss some evidence about visuomotor integration, providing clues from the Poffenberger paradigm, and again combining behavioral, functional, and structural evidence. The Poffenberger paradigm is an ingenious way to test visuomotor integration and interhemispheric transmission of information. It is a simple reaction time task. In the cross condition, a dot is presented in one visual field, in this case the left visual field, and the subject is asked to respond with a contralateral hand, the right hand in this case. Whereas in the uncrossed condition, the visual stimulus and the response hand are in the same side. Reaction times in the cross condition are typically slower than in the uncrossed condition. This is because in the uncrossed condition, the visual input and the motor output involve the same hemisphere, whereas in the cross condition, the visual information has to be transferred to the motor areas of the opposite hemisphere to control the manual response. Therefore, it is commonly believed that this difference in reaction times provides an index of interhemispheric transfer time of visuomotor information through the corpus callosum. We have adapted this design to test blindsight patient GY with right hemianopia. Therefore, stimuli presented in the right visual field are unseen, whereas stimuli presented in the left visual field are consciously perceived. This slide reports reaction times, behavioral results in the patient GY. Essentially, when the stimuli are projected in the left intact field, reaction times are typically slower in the cross condition compared to the uncross condition, which essentially replicates previous findings in healthy observers. However, when the stimuli are projected in the blind field, reaction times in the uncrossed condition are paradoxically faster than in the cross condition. This seems to suggest an active role of the intact hemisphere in sustaining visuomotor integration for unseen stimuli. We have proposed that when the stimulus is projected in the blind field and initially processed by spared excess striated area in V1 damaged hemisphere, this processing requires the contribution of homologous visual areas in the intact hemisphere, which are recruited through the callosum. Now, when the manual response is controlled by the intact hemisphere, as in the case of the cross condition, only one interhemispheric transfer is required. However, when the manual response is controlled by the V1 damaged hemisphere, as in the case of the uncrossed condition, then a second transfer of information is required from extrastriate visual areas in the intact hemisphere to motor areas in the V1 damaged hemisphere. And this will explain why reaction times are paradoxically slower in the uncrossed condition when the stimulus is projected in a blind field. We first compared fMRI activity to non-conscious versus conscious perception of stimuli, irrespective of response hand. Basically, we found bilateral activity in extrastriate areas in the dorsal stream, including the intraparietal sulcus, the supramarginal gyrus, the cuneus, and the posterior part of the corpus callosum that integrates information between the parietal lobes. This picture suggests an increased need for interhemispheric communication between dorsal stream areas specific for non-conscious visually guided manual action, which seems in keeping with our initial interpretation of reaction time results. Next, we compared activity in the crossed versus uncrossed conditions during non-conscious perception, therefore when the stimuli were projected in the right blind field. And we found that uncrossed rather than the crossed responses produce greater bilateral activity, essentially activating the dorsal premotor and supplementary motor areas, and also the anterior part of the corpus callosum that connects these motor areas in the two hemispheres. So overall, it seems that there is more than a simple relay of visual information to motor areas within the damaged hemisphere that seems to require, as initially predicted, the contribution of the corresponding motor areas in the intact right hemisphere. Next, we analyze the pathways of information flow during non-conscious perception with ROIs placed in dorsal stream areas and in premotor areas in the crossed conditions displayed here in red and during the uncrossed conditions displayed in blue. essentially, we found that there is correlation in activity of extrastriate dorsal areas, irrespective of the response hand used. Then that there is a correlation between visual and motor areas in the intact hemisphere in both crossed and uncrossed condition, but this communication between visual and motor area seems to be impaired in the V1 damaged hemisphere. And then finally, there is a correlation of fMRI activity between motor areas in the intact and V1 damaged hemisphere, but only when the damaged hemisphere controls the response. So overall, it seems that the integration between visual and motor areas in the V1 damaged hemisphere is impaired and requires the sustain of the intact hemisphere. We next analyze the topology and geometry of the callosum through connectivity-based parcellation. Each sphere represents the center of gravity of each control subject and the corresponding callosal segment based on different colors. Whereas the center of gravity of each callosal component in GY is represented by the square. And as you can see, there is a preserved topology of the different callosal segmentation and geometry, and no specific alteration in the comparison between controls and GY. However, when we analyze the number of fiber tracks composing each callosal section, we found a significant increase in homotopic connections linking posterior parietal areas, again suggesting that in GY there is an increased exchange of visual information between dorsal stream areas of the two hemispheres that sustains visuomotor integration for unseen stimuli. Finally, I'd like to thank again Marta Garrido for organizing the symposium, Sarah, Gina, and James Bourne for their insightful presentation, you all for the attentions, and the funding agencies that supported this line of research over the years. Thank you again.